Hello, my name is Robert Riehn. I'm a faculty member here at NC State, and I have been at NC State since 2006. My specific research area is biophysics. Within biophysics, I have fairly diverse interests. My first large interest is genome organization, protein-DNA interactions, and the physics that are involved with having polymers interact under confinement. A second large research area is developing nanotechniques for bioanalytics. Those would be nanofluidics, or they would be nanophotonics, things of this nature. A newer project is using artificial intelligence for the analysis of interacting insect populations. As part of all of these fields, we often encounter interesting statistic mechanics. Specifically, those fields of statistical mechanics that come up are the transitions between a thermal description, where temperature matters, and an athermal description, where momentum and velocity matter more. A second theme that has often come up is the transition from a regime where things are in equilibrium to a regime where things are not in equilibrium. Within a non-equilibrium regime, we again distinguish between something that is far out of equilibrium and something that is close to equilibrium. And we find that things really change as we transition between these different areas. These fields may seem very widely spread, but they follow a common thread or a common thought. And this thought is the following. The real world around us, and especially living things, are very complex. We measure a system that has many different degrees of freedom, and in order to understand the system, we have to reduce the amount of data. So we have to find collective variables, and that is at the center of my research. We have to take, for instance, a movie, and we have to find a way of analyzing this movie that is practical, that is robust, and that makes it easy to link what we see in the movie to our microscopic model of how the system works. Finally, once we have found this description, then we try to see whether our understanding or macroscopic understanding of the system is consistent with the macroscopic behavior of that we see in the experiment. We are now in the lab. The lab consists of multiple different rooms where the different functions are performed. We have laboratories for assembling devices, doing some chemistry, nanophotonics, nanofluidics, some general electronics, and for molecular biology. We are now in the laboratory where we assemble our nanofluidic devices. This here would be one of our nanofluidic devices, where you can see the 4 ports where liquid enters the device. The actual channels you cannot see. In this room we take wafers that have been prepared within a semiconductor clean room here on campus, and we seal the device, we prepare it so it can run it, we load it onto a sample holder. We are now in the biolab. In the biolab we have an autoclave, freezers, fridges, centrifuges, water baths. Those are the kind of things that you need in order to prepare biological samples that we then analyze in our nanophotonic or nanofluidic devices. This is a room within which we do chemical processing. Chemical processing typically is performed within the context of wafer preparation and device preparation. We have a reasonable capacity here to perform basic chemistry. Here we have an optical laboratory with a single molecule microscope. This is really the heart of where most of the data of my research group is taken. The setup is rather unique. We have 4 different lasers. We can steer where the laser hits the sample. We have assembled our own control electronics, our own electronics for controlling pressures. The software is home written. So it's a relatively special instrument. Here a graduate student has mounted a nanofluidic device underneath this plastic block. Green tubing here that you can see is tubing that is used to push liquid and DNA around within the device. The microscope objective illuminates everything from the bottom. Everything is controlled by our controller that we built in-house. Down here we have the most expensive bit of the whole setup. This is a camera that costs something like $30,000–$40,000 that can see single photons. We have here an image splitter that is able to take different color channels apart. And this board that you see now is our laser board, where we have 4 different lasers that get combined to be sent into the microscope. This room houses a new experiment in nanophotonics that a graduate student has assembled over the last 3 years. It is essentially a near-field optical interferometer that we will use to detect single proteins or other biomolecules. Students in my group have typically graduated something like 5 to 6 years, probably 5 years. We had students that graduated in 4 years. Typically students take 1 year of classes, maybe 1.5 years of classes. We have the students perform research for about 3 years, and then depending on the student's skill in writing, it may take half year to 1 year to write the thesis. My group typically has 2 to 3 students, and I attempt to interact with students 2-3 times a week at least. Students from my group have gone on to postdoctoral appointments. They have gone to companies that specifically were interested in a student's skills in bioanalytics. So for instance, somebody has gone to a sequencing company, somebody has gone to a microfluidics company. Students have gone into software companies where they were able to leverage all the software and computing skills that they gained in my group. And finally, 2 students have joined consulting companies. During a time in my research group, you gather a lot of skills that may not seem directly related to biophysics. So the first thing that people learn is nanotechnology for bioanalytics. They learn about instrumentation, microscopy, optics, nanofabrication, biological preparation techniques. The second large field is image and data analysis that I had already talked about. Quite a few students in my group have done considerable amounts of computational works and simulations. Those could be using optics packages, or those could be in molecular dynamics simulations. Finally, students learn the specific physics that underlies their specific project. In addition to these technical skills, I expect students to write 2 or 3 first author papers throughout their PhD. I expect students to present at national and international conferences. And students obviously write a thesis.